Let me record. All right, let me just go back to the other page so everyone's up to speed. All right, so hello everybody. This is our schedule for this week and next week. Some of you may have noticed on the calendar, it was showing May 25th for the final exams. But one of your colleagues noticed that, mentioned to me that it was on, it was on May 25th. It should be on June 1st. So, which is the way it is on the actual syllabus. So kind of keep that in mind. Both exams are on June 1st. This is our schedule for this week. And so today we're going to start with the urinary system. All right, a little ragged opening there, but we'll get going. All right, so urinary system. So as you can see, this is a picture showing the abdominal cavity with some of the organs removed, but here you can see the kidney. So we're going to focus with the urinary system on the kidney, what's called the ureter, the bladder, and then the urethra. Those are really the four main organs of the urinary system. And you can see them right here. So the bulk of our discussion is going to be on the kidney, because the kidney is really the factory of the urinary system. This is where blood is processed, filtered, modified, and essentially urine is formed. The other structures are not any less important, but their roles are different. For example, the ureter is responsible for transporting urine from the kidney to the bladder. And of course, there's two of them because there's two kidneys. Once the urine reaches the bladder is a very muscular organ, as you'll learn. The urine is stored here prior to elimination. During elimination or urination, urine is eliminated through what's known as the urethra to the outside of the body. So those are the four structures. Like I said, we're really going to focus really on the kidney. This is a gross picture of the kidney. You can see, of course, it's shaped like a bean. Couple structures you can see from the get-go is that the kidney is covered in a capsule, kind of a connective tissue capsule. We also see this area right here, which is known as the hilus, which is very similar to the hilus of the lung. Remember, the hilus of the lung is where we have the blood vessels entering and leaving, as well as the bronchial tubes entering. Well, in this particular case, we have blood vessels entering and leaving, arteries and veins, and then we also have the ureter leaving. So that's all in what's called the hilus. What does the kidney do? Well, we often think of the kidney as an excretory organ, right, involved in making urine, and that's true. But its role is much more complex than that, because what it needs to do to form urine are several different steps. First of all, it plays a role in maintaining the chemical consistency of the blood, regulating what's going to move from the kidney back into the blood, regulating what ions, how much water, glucose, things like that. So it basically kind of maintains the consistency of the blood. It helps to regulate the volume of blood plasma, blood pressure, as well as pH, or the acidity of the blood. And we're going to talk a bit about how it does that. So in the process of regulating what is being filtered, what is in the urine, what's in the blood, it's now really preparing for what's going to be excreted. And what is actually excreted is primarily toxins, metabolic waste, and excess water, right? And the amount of water that's excreted really depends upon how much we've consumed. If we haven't consumed much of the water remains in the body to prevent us from being dehydrated. All right, there's when we talk about waste products or metabolic waste and things like that, so besides the water and the toxins, some of the things that are excreted are what we call nitrogenous compounds. Urea, uric acid, and creatinine. These are all nitrogenous compounds. Some of them are really some breakdown products of proteins. The most abundant is urea. That's the most prominent one. Interestingly, if you're looking at a kind of a comparative anatomy approach, the different animals excrete differently, and mammals typically excrete urea, but it's interesting that birds and some reptiles excrete uric acid more so, whereas fish excrete actually ammonia. But we excrete primarily urea as well as the other two. All right, so where is the kidney located and what do they look like? Well, they have kind of a reddish brown color. They're located in the retroperitoneal space. You remember we spoke about that last time, the retroperitoneal space. This was this area that's behind the peritoneal cavity. Remember I mentioned things like the duodenum. Parts of the colon are found there, as well as the pancreas. Well, so is the kidney. Just to kind of give you an idea of its proximity to the vertebrae, it's basically between the T12 and L3 region of the bird. So you can kind of have an idea of, especially with boxing, you know, it's illegal to punch in certain areas, and that's one of them, just due to the potential for kidney damage. So the kidney is about 12 centimeters tall, where if you think about a centimeter is 2.5 inches, you're looking at about 5 inches is the size of an average kidney. As I mentioned, we have this structure known as the hilum, where we have blood vessels that you can see. We also have the ureter. And one thing that's not showing is we have nerves that are traveling into and out of the kidney. This actually happens to be a baby's kidney, which is really not fully developed. All right, so this is a transverse view showing where the kidneys are located. So this is the anterior side. This is the posterior side. Here's the vertebrae. This, what you see in white, this is the peritoneal membranes. And this area back here where we have the kidneys, also the aorta and the inferior vena cava, this is all the retroperitoneal region. All right, so we saw what the kidney looks like on the surface, but what about the inside? If you were to slice into the kidney, what you would see are a couple things that I'm going to point out. First of all, of course, we have this outer capsule, which covers the kidney. Underneath that, you can see we have these regions that look different from each other. We have this region out here, and we have this region in here. This outer region is known as the renal cortex. This inner region is known as the renal medulla. And if you look carefully, you can see these structures that I'm outlining. These are called pyramids for obvious reasons. They're shaped like a pyramid. So what we can say is that the renal medulla is made up of individual segments or structures called pyramids. There's a bunch of regions. I'm not going to go over all of them, but what I am going to show you is kind of the path of urine flow. And as urine is formed, it travels from the cortex to the medulla into these little openings that I'm pointing out right here that are known as the minor calyces. Each of them is called a minor calyx. And each minor calyx empties into what's known as the major calyx. So this is area. So these are all minor calyces, and they empty into the major calyx. Then this empties into what's called the renal pelvis, which then drains into the ureter. So that's really the path. Cortex, medulla, calyces, pelvis, ureter. There's a lot more to the kidney. We're going to get into some of the microscopic aspects, but that's kind of just at the more of the superficial level. All right. So like I mentioned, we have the renal cortex. We have the renal medulla, which is made up of these pyramids. And then we have the minor and major calyces, and then which empty into the renal pelvis. Now, a little bit about the vasculature. There's a lot to the kidney as far as blood vasculature, and it's not on the slide here, but just to let you know, about 20% of the overall cardiac output that goes through the body goes through the kidney. So about 1/5 of all the blood that goes through the heart, that circulates through the body passes through the kidney. So it's a very important structure, obviously, for filtering. As far as blood vessels, we have the renal artery, which is a branch off the aorta. And this branches into a couple smaller arteries. You can't see them right here. I'll show you in more detail later. They branch into smaller and smaller vessels, the segmental, interlobar, and arcuates, as well as some others. But the one that enters into the kidney is the renal artery. As far as the drainage, it's the renal vein, which collects blood from several other smaller veins. And I'm going to show you those on some of the upcoming slides. All right. So what we've seen so far with the kidney is the superficial view, right? We've got the cortex, the medulla, the pelvis, right, and then the ureter. But if we look microscopically in the kidney, remember this is an organ that's involved in maintaining the chemical constituency of the blood, maintaining, what's going to be in the urine, what's going to be in the blood flow, things like that. So what is it about the structure of the kidney that allows that to happen? Okay. Well, the functional unit of the kidney is what's called the nephron. And there are at least 1 million nephrons within each of these kidney that are scattered or really packed through the cortex and into the medulla. The nephron is the functional unit, meaning that the nephron is the structure that actually helps to filter blood, maintain the constituency of the blood, all those other functions that I mentioned before. What we're looking at right here are really two nephrons. These are blood vessels. This structure that I'm circulating, that I'm kind of forming a boundary around, this structure all throughout here, this is one nephron. And this structure up here is another nephron. Each nephron consists of two main components, two main parts. This area right here, which is known as the renal corpuscle. Within the renal corpuscle, we can contain a capillary bed, which you can see better up close. So these two are the same regions. This right here is just covered. This has the cover pulled off. Within the renal corpuscle, we have capillaries known as the glomerulus. Right there. Surrounding the capillary, we have what's called the glomerular capsule, which consists of a layer, what's called the parietal layer, and podocytes. You can't see them at this level, but I just want to let you know that we have the glomerulus and the glomerular capsule. If you want to think of, for example, what it kind of looks like, if you think of the capsule as like this, the glomerulus is like this. So the glomerulus is like a fist with inside the C. All right. What about these tubular structures? Well, that's in fact what they're called. They're called tubules. And we have two sets of tubules. We have what's called the proximal convoluted tubule, which is directly connected to the glomerular capsule. You'll see this on some other slides. It's called proximal because it's close to the capsule. It's called convoluted, as you can see, for obvious reasons, right? So this is the proximal convoluted tubule. This connects into this hairpin-looking structure. Here's one here. Here's this area right there. This is known as the, well, actually, this area, as well as others, is known as the distal tubule. I actually did leave something out. And then let me go back for a second. Apologize for that. Let me go back here for a second. I left one other thing out. Let's see. Otherwise known as nephron loop. I know I left something out. There we go. Sorry about that. Let's see. Let me back up for a second. Okay. So what do we have? So we've got the start from scratch. We've got the glomerulus, the capsule. We've got the proximal tubule, which is kind of coiled. Then we've got this hairpin structure, which is the loop of Henle, or the nephron loop. You can see it right here, as well as down here. And then that loop of Henle continues into another convoluted tubule called the distal tubule. And that finally empties into what's known as the collecting duct, which will eventually drain this into the renal pelvis and out. So if you want to kind of monitor the flow of urine, the urine starts, the blood, as we'll see, first from the circulation enters into the glomerulus. It's filtered. The plasma or whatever material is going in goes through the proximal tubule, then through the loop of Henle, the distal tubule, and whatever is left ends up being excreted. Not everything that goes into the kidney is actually excreted. Actually, about 99% of what passes into the kidney gets reabsorbed back into the blood, which is important because that's how we salvage all of our electrolytes, much of our ions, our nutrients, water, things like that. All right, so this is a photomicrograph of a nephron, or at least kind of it's maybe hard to sort of navigate, but what we're looking at right here, this is the glomerulus. Just a kind of a collection of a coiled capillary bed. This area right around here is Bowman's capsule, or otherwise known as the glomerular capsule. These structures that you see kind of in red, these coiled structures, this right here, as well as all these others, these are all proximal tubules. And what you see kind of looks a little bit different. This kind of a shorter cell, right? Comparing this to this, this is a proximal tubule. This is a distal tubule. Distal tubule is a bit lighter and thinner. So it's kind of hard to kind of navigate, but basically the blood gets filtered into the proximal tubules. It looks scattered like this because it's all coiled. And if we go back to this previous slide, you can see that all these tubules are just scattered, all kind of coiled through. So just a little bit of histology: glomerulus, capsule, proximal tubule, distal tubule. This is a better view looking at the loop of Henle. The loop of Henle is, or the nephron loop is very thin. Different view. And from the loop of Henle, then it drains into the collecting duct. So you can kind of see the change in appearance from these very tall looking cells, right? There's flatter cells with the distal tubule, flatter cells with the loop of Henle, and even flat cells with the collecting ducts. So the reason for, you might wonder, why are the proximal tubule cells, why is it so thick? Why is it kind of like a fuzzy appearance? That's because they have a lot of microvilli. Most of what is going to be reabsorbed from the kidney back into the blood occurs in the proximal tubule. So a lot of water, a lot of salts, once they go into the tubule, they're reabsorbed back into the blood. Okay. So those are the main structures of the nephron, right? I spoke about the glomerular capsule, which is the glomerulus plus the capsule, which makes up the corpuscle, the glomerular corpuscle. This area. We also have the tubules, right? The distal, the tubule, the proximal tubule, the collecting duct, and the loop of Henle. I wanted to mention a couple other features that are kind of located around the renal corpuscle, and this is what's called the juxtaglomerular complex. So if this is the glomerulus, we have these interesting groups of cells that are scattered. We have, for example, these little purple-looking cells. This, by the way, is the distal tubule. These purple cells are what are called the macula densa, right? We also have what are known as granular cells, which you can see right along here. And we also have what are called mesangial cells, which are kind of hard to see, but they're located here. I'm not going to have any pictures asking you to identify where they are in the picture, but you should know there are 3 other cells, groups of cells found in the kidney that make up what's called the juxtaglomerular complex. These cells all play a very important role in maintaining blood pressure. The granular cells produce an enzyme called renin in response to low blood pressure. So if blood pressure coming into the kidneys is low, renin is produced by the granular cells, which helps to increase blood pressure. In physiology, we actually go over how it does it, but it helps to increase blood pressure. We also have the macula densa cells, which are located in the tubule, and they respond to, they're kind of like a chemoreceptor. We've learned a bit about chemoreceptors, like with the respiratory system. These are receptors or cells that sense the number of ions, specifically sodium, that are traveling through the kidney. If the sodium levels are too high, that's an indication that the blood pressure is too high, and the macula densa responds by producing a substance that will lower blood pressure. So it's kind of neat. We have this very well orchestrated regulatory mechanism which helps to respond to when the blood pressure is too low or too high. And we have these mesangial cells which also regulates blood flow. So there's a very intimate association between the cardiovascular system and the kidney. Which explains why some of you may have, for example, may have had grandparents or relatives or just really anybody that has cardiovascular problems, and quite often that translates into kidney problems. Or in contrast, kidney problems will translate into cardiovascular. So there's a very intimate association between those two systems. All right. So what does the kidney do? What are the functions? Well, we already know the big picture. We already know that the kidney's role is to maintain the chemical composition of the blood. At the same time, it's going to pick out certain substances that are in blood that we don't want to keep, and it's going to get rid of them. And it's also going to take some of the constituents of blood and it's going to move them back into the blood from the kidney. So it does this by 3 distinct mechanisms. First one is called filtration. We take a look at the flow of blood into the kidney. So this is kind of a close-up view. This is really the glomerular capsule, and this would be some of the tubules. Blood enters into the kidney and is filtered through what's called the afferent arteriole. The afferent arteriole brings blood into the kidney, into the glomerulus, where it's filtered. One way to think about filtering, think of like a piece of cheesecloth. Something gets filtered across, so the blood, right, plasma leaks out of the glomerulus through a filtration system and enters into the kidney tubule. This is known as filtration. And this is essentially the plasma, the fluid portion of the blood. Once the plasma is in the kidney tubule, the kidney ends up realizing, well, gosh, a lot of what's here, we don't want to get rid of. It needs to go back into the blood. And this is where we do what's called resorption or reabsorption. You can call it resorption, where water, nutrients, salts, things that have been filtered end up going back into the blood. So those are conserved. Like I said, about 99% of what goes into the kidney returns back to the blood. Now, occasionally we have substances that weren't filtered at the glomerulus, but still end up in the kidney by a different mechanism. This is called secretion, tubular secretion. And you might wonder, it's like, well, how are these 2 different? How is 1 and 3 different? With step 1, the filtration, what goes into the kidney are things like water, salts, sugars, nutrients, things like that. What enters into the tubule by way of secretion are toxins, metabolic waste. If you've taken any medication, like antibiotics, the medication breaks down, generates toxic substances that need to be eliminated. So they're typically eliminated in this fashion. At the end of secretion, what's left now, the byproduct is urine. So it's kind of like a formula, right? Filtration comes in, we subtract out the reabsorption, add in the secretion, and then we have our urine. All right, so this is really a summary. Filtration, resorption, secretion. Those are the 3 steps involved in forming urine, helping to also maintain the constituency of the blood. All right. So let's take a little look at filtration. This is another view of our nephrons. This is a nephron. This is a nephron. And if you kind of navigate, you can see there's the glomerulus, there's the corpuscle. Here's these tubules in yellow, right? Eventually leading out into what's known as a collecting duct. Here we have 2 distinct nephrons. I might add there are a couple different types of nephrons. One hangs out mostly in the cortex. This is called a cortical nephron. One extends into the medulla. This is known as the cortico or a juxtamedullary nephron. Still the same, still does the same stuff, but they're just located in different areas. Cortical nephron and juxtamedullary nephron. All right, so blood is filtered. So just keeping track, blood enters into the glomerular capsule through this vessel known as the afferent arteriole. The afferent arteriole connects and moves into the glomerulus, and then the blood is filtered. and eventually goes into the proximal tubule. Now, this vessel, known as the efferent arteriole, contains material in the blood that wasn't filtered. So what is typically not filtered? Large molecules like proteins, even cells, blood cells, don't normally get filtered unless you've got like a kidney infection or some other condition. But what goes into the kidney are, again, water, salts, sugars, and then large proteins, stuff like that, remains in the blood and then goes back into the afferent arteriole. We're going to go through the details about this. Now, you might wonder what this is a new term, Bowman's capsule, right? Well, as you know with anatomy, there's always more than one name for the same thing. The old name was Bowman's capsule. It's now known as the glomerular capsule. All right. So let's talk a bit about filtration to understand some of the details of it, because this is actually very similar to something that we see with the diffusion of oxygen in the lungs. We have this filtration of material from the blood into the kidney. So this is our afferent arteriole, blood's coming in. Here we have the glomerulus, and here we have the proximal tubule. Well, a couple things to measure. First of all, you'll notice that we have this kind of weird looking yellow spidery stuff covering the glomerulus. It doesn't look like a blood vessel. Well, that's because what you're looking at in yellow are specialized cells known as podocytes. If you look carefully, this is a nucleus, this is the cell, and we have these little extensions that are sometimes called foot processes, kind of like an astrocyte. If you remember how astrocyte wrapped around the capillaries in the blood to help maintain the blood-brain barrier, we have these podocytes that wrap around the glomerulus. They are part of what we call the filtration process. So as blood enters into the kidney from the afferent arteriole, as it passes through the capillary, which by the way happens to be a fenestrated capillary, which little pores in it, it then has to pass through another layer called the podocyte and what's known as the basement membrane. So before it gets into the proximal tubule. So what does that mean? That means what we call the filtration membrane. For blood to go from the arterioles filtered into the proximal tubule, it has to pass through the fenestrated capillaries, through the basement membrane, which you can't see right here, and then pass through these slits that are found in the podocytes. So let's take a closer view of that. This is really what we're talking about here. This is our capillary. These are the fenestrations. Once the fluid passes through the fenestration, excuse me. Once it passes through the fenestrations, it has to pass through these little slits of the podocytes, which also are where we have the basement membrane. Now, by the time we pass through the filtration slits, we have now what we call our filtrate, or what's going to go into the tubule. And that's going to include things like water, ions, sodium, potassium, calcium, things like that, glucose, amino acids, right? Urea. So you notice that proteins aren't there. That's because proteins are too big. They can't get through this slit. So say if you have blood work, or I should say a urinalysis, just routine, or maybe doctors are suspecting something and there's protein, a lot of protein in your urine, that could be an indication of maybe the minimal, maybe a very slight kidney infection, or it could mean something else as well. So, because normally you will not find much in the way of, proteins in the blood. All right. So speaking of filtration, this is probably a better indication, better view of how blood flows through. This would be the capillary, or really the glomerulus. the plasma, remember the fluid portion, right? Because the cells, blood cells can't get through, or proteins. They're going to pass through these fenestrations. They're going to pass through, this is the basement membrane. And they're going to pass then through these what are called filtration foot processes or filtration slits. By the time it gets through here, now we have our filtrate. So it's kind of like really a triple filtration system consisting of the capillary, the basement membrane, and the podocytes. All right. Now, getting more into the vasculature, I kind of have been teasing you all along, talking about how, yes, the kidney has an excellent blood supply. 20% of the cardiac output goes through the kidney. It's a lot. And I've mentioned a couple vessels, but now I'm going to give you the entire extent of the blood flow through the kidney, so you can really see the significance of it. And many of these are vessels well, a few of them are vessels you've heard of, but so many of these are new. So let's break this into two parts. Let's break this into the blood supply and then the drainage. So the blood supply consists of the renal artery, which branches off the aorta. There's the renal artery. The renal artery then splits into what's known as the segmental artery, which splits further into what's called the interlobar artery. And you can kind of follow along on this picture, which splits further or branches into the arcuate. And the way I've used to kind of remember that, I think of arcuate like an arc. The arcuate artery kind of arcs in this direction. From the arcuate artery, we then have the cortical radiate artery, which is very small to see up here, eventually into the afferent arteriole, which you guys remember. Remember, that's this. And there's the efferent. From the afferent arteriole, we then drain into the glomerulus, where the blood is then filtered. Whatever is not filtered continues into the efferent arteriole, into vessels known as the peritubular capillaries and the vasa recta. You can actually, well, I'll show you later, that you can see some pictures of them. Eventually into the cortical radiate vein, arcuate vein, interlobar vein, renal vein, and then out through the IVC. You can see through many of these, the arteries and veins parallel in their names: renal, renal, interlobar, interlobar, arcuate, arcuate, cortical radiate. The only difference is we have a segmental artery, but we don't have a segmental vein. All right. So I mentioned before that from the efferent arteriole, whatever has not been filtered initially passes into what we call the peritubular capillaries and the vasa recta. You can go back here, these right here. So what do these look like? These are vessels that basically are wrapped around the tubules. What you see in kind of a purple or violet color. And what happens here is not only does the blood that has actually not been filtered, that contains blood from the efferent arteriole, but this is also going to be where a lot of the material that's in the tubules is going to be eventually be reabsorbed. And all this eventually collects and drains out through the renal vein. So the vasa recta, which is what we find in the juxtamedullary nephrons, and the peritubular capillaries is what we find in the cortical nephrons. So you're kind of getting a crash course here in your urinary system anatomy. All right. So a little bit more detail about so now we know all the vessels, but I want to just talk a little bit more detail about them because it's a very important part of kidney function. As we know, of course, and kind of summarize some things, the glomerulus, or filtering across the glomerulus and these podocytes right here, produces a filtrate, right? It squirts over into the proximal tubule, and then combine that with the reabsorption and secretion, we're going to end up with urine, right? What enters into the glomerulus, of course, is the afferent arteriole. What drains the glomerulus is the efferent arteriole. Afferent arteriole is larger in diameter. This is very important. It helps to maintain the blood flow. Just to give you an idea of how much fluid is generated, about 1 liter of fluid is generated every 8 minutes. So that's about 7 liters per hour, right? That's what, about 170 liters per day. 99% of that is reabsorbed. You can imagine if we had 170 liters that we had to pee out, we'd be on the toilet all day long. So fortunately, most of it's reabsorbed to conserve a lot of the water, a lot of the solutes. These peritubular capillaries, of course, they're all, I don't know what absorption is, but they're all adapted for absorption. So a lot of the material, remember, that is filtered, most of it's filtered, ends up going back into the blood, and it goes back into the blood through the cortical nephrons, through the peritubular capillaries, and much of which is secreted, is also secreted from those vasa recta and peritubular capillary. So let me back up for a second, because I was kind of little kind of convoluted here. So these peritubular capillaries, which continue from the efferent arteriole, right? These are the capillaries in which any filtrate from the kidney is going to be reabsorbed back into the blood. It's also the place where any material that was not filtered, which needs to be secreted, things like antibiotics, other types of waste, enters into the kidney through those vessels. All right. So the filtrate, a lot of this is just review. The filtrate proceeds from the tubules, from the proximal tubule into the loop of Henle, into the distal tubule, where eventually it goes into the collecting duct. So proximal tubule, loop of Henle, distal tubule, and then this long, very straight structure is the collecting duct, and several different nephrons drain into that. This is probably the most, even though all along the kidney, the tubules are important in secretion, absorption, all those things, the collecting duct is probably one of the most important areas in really conserving water. The collecting duct is under the control of a hormone that's stored in the pituitary called ADH, antidiuretic hormone. And what happens is, as the filtrate passes through the tubules, much of it, as I mentioned, is reabsorbed back into the vasa recta and in the peritubular capillaries, and then some from the vasa recta and the capillaries are secreted. But by the time we get to the collecting duct, there's still some water. So we want to kind of finalize the process. We want to move whatever water we need in our bodies back into the blood. And the hormone ADH acts on the collecting ducts to make the collecting ducts more permeable, so we can move water from the collecting ducts back into the blood. So antidiuretic hormone is the hormone of interest here. All right, so that's really the bulk of the lecture, right? The kidneys, as you can see. That's really where all the action takes place. That's really the factory, right? That's where blood is being filtered, and we're modifying what's being filtered, eventually coming up with a product of urine that contains waste, some water. Most of what was in it before, of course, is reabsorbed back into the blood, which has been conserved. Once the urine leaves the kidney, it passes through the ureter. And this is a section, a photomicrograph of the ureter. showing some of the histology. The histology of the ureter consists of a mucosa and a muscularis and an adventitia. So some of it, those names might sound familiar to you. The mucosa consists primarily of transitional epithelium. You remember that term transitional epithelium? We spoke about it in the bladder early on in the semester, but it's also found in the ureter. That transitional epithelium is also surrounded by two layers of muscle. And the muscle is important for contracting to help to kind of move the urine along, although a lot of it just moves by way of gravity, but it does help to move it along. And then that's surrounded by a layer of adventitia, a type of connective tissue similar to a serosa, but it's different. So this is the lumen of the ureter. So the urine moves from the kidney through the ureter to the bladder. The ureter is shaped such that it prevents there's not normally a backflow of urine. It makes sure it's shaped so that it's kind of a continuous flow. Carries the urine into the bladder, and here we have the bladder. All right, and there's the ureter, there's the bladder. Here we have the urethra. This is what a full bladder looks like. It's a very thick, muscular sac that stores urine, which eventually is going to be eliminated. So let's take a look at the bladder, a little bit more detail. Here's the bladder, here are the ureters. First and foremost, I mentioned muscle. The bladder has a very thick, smooth muscle known as the detrusor muscle. That's this thick muscular wall, okay? There's folds in the bladder called rugae. And remember from the stomach, when the stomach is folded, when it's empty, you can see these submucosal folds called rugae. Bladder is similar in the sense that when it's empty, you can really see those rugae. When it's stretched out, when it's full, you can't as well. All right, some other features of the bladder that are important. So the bladder is stored here in the main component. We have a couple sets of sphincters. We have what's known as the internal urethral sphincter and the external urethral sphincter. And remember, just kind of going back, we spoke about the esophagus. Remember having the upper esophageal sphincter and the lower esophageal sphincter. And remember with the digestive system, we had the internal anal sphincter and the external anal sphincter. Well, this is similar to the anal sphincters in the sense that the internal urethral sphincter consists of smooth muscle and it's under autonomic control. The external urethral sphincter is voluntary and it consists of skeletal muscle. So very similar to the anal sphincter, that when you have the sensation that you've got to go to the bathroom, this puts pressure and you'll start to see the relaxation of the internal sphincters. And then when you make that willful decision to go to the bathroom, you're in on the toilet, that external urethral sphincter voluntarily will relax. So very similar to the concept that we saw with the anal sphincters. All right. So urinary bladder, like I mentioned, as a muscular wall. Not only does it have a muscular wall, it has an epithelium, which is transitional, much like the ureter, and it has an outer adventitia. So the similarities are between the ureter and the bladder are the transitional epithelium and the adventitia. The intervening layer is different. Even though the ureter has some muscle, it's nothing like this very thick detrusor muscle. The significance of that, if you think of structure and function, the bladder needs to stretch and then contract to expel the urine. So it's under a lot of pressure. So we need to have that thick muscle. All right, so what about the urethra? So this is where the urine leaves when we're urinating, leaves the bladder, goes in the urethra. The urethra also has transitional epithelium, at least in the beginning. But as we get close to the exit point, we have actually a couple things. We have, for example, stratified and pseudostratified columnar in the males. But at the very end, in both males and females, you don't see it right here, is non-keratinized stratified squamous. So here's the kicker. So in both males and females, it's transitional in the beginning. At the end, it's non-keratinized stratified squamous in both. In the middle, for some reason in males, we have columnar. Obviously, there's something going on there, whether it's secretions. All right. Now, the sphincters that I spoke about, of course, are technically considered part of the urethra. So we have the involuntary internal sphincter and the voluntary external sphincter. All right. So we're almost at the end. Let's talk a bit about what's known as micturition, which is the same thing as urination. This is also known as voiding or urination. This is under the control of both the autonomic and the somatic pathways. And if you think about it, it kind of makes sense, right? We have smooth muscle with the internal sphincter and also the detrusor. Those are controlled by the autonomic. And then we've got skeletal muscle with the external sphincter that's controlled by the somatic, all right? So let's take a look here. And I know this is a lot of information, but let's kind of navigate ourselves. Here we've got the bladder, and here we've got the two sphincters. But what we have in addition right here are nerves and the spinal cord and the brain, okay? So let's take a look at the left side right here. So what happens as the bladder, obviously the need to urinate depends upon how full the bladder is, right? So the bladder distends or stretches as it's fulfilled. As it stretches, basically it sends impulses to the spinal cord and the brain. Okay, basically up this way. Yeah, here we go. In response, so we have stretch receptors activate neurons that are going up to the spinal cord as well as the brain, primarily the pons area. Once the neurons reach there, this activates the parasympathetic pathway, which comes down, and this does two things. This stimulates contraction of the detrusor muscle, and it also triggers relaxation of the internal sphincter. So you see this kind of a purple neuron? So stretch receptors activate this neuron, which goes up the sacral portion of the spinal cord, up through the pelvic nerve, up to the pons, back down through the spinal cord by way of the parasympathetic system through this purple nerve, innervates the detrusor muscle, stimulates contraction, which is going to squeeze the urine out through the internal urethral sphincter, which is now relaxed. So contraction and relaxation by way of the autonomic nervous system. And you can see that right here. Parasympathetic efferent stimulate contraction of the detrusor and open the internal sphincter. Now, what about the external sphincter? Okay. Well, normally what happens is there's a somatic neuron that comes through that typically keeps the external sphincter contracted. It keeps in a constant state of contraction. In order for us to urinate, this sphincter has to be relaxed. And so somatic neurons there are eventually inhibited, which allows the external sphincters to relax. All right, so let's kind of go over this. This is kind of a loaded section right here. So we got this full bladder, activates stretch receptors in the detrusor muscle in the bladder wall, sends nerve signals up through the sacral portion of the spinal cord, initially all the way up into the pons. From the pons, the neurons then come down via parasympathetic pathway, innervating both the detrusor muscle, causing the muscle to contract, cause the internal sphincter to relax. The external sphincter is still in a contracted state, right, thanks to the somatic neurons, but these neurons are now inhibited, which frees the muscle to now relax. This is a learned response. So when you talk about kids being potty trained or not potty trained, when they're not potty trained, basically what happens is we don't have this inhibition to begin with, right? Basically, it's constantly open. We need to eventually, and so the part of the regulation, the development, is to have the ability to tonically control the external sphincter, because in kids, when they gotta go, right, they can't stop it. All right, this is actually another video. It's a bit too, I'm a bit involved, but you can kind of see some of the details. If it is timely to urinate, signal sees an external urethral sphincter relaxes. So here's this external sphincter relaxing. All right, let me show you this quick video showing the process of micturition. Let's see if this works. Let's see. Hopefully it'll work. Let's see. Let me just pause for a second. Hold on a second. I'm going to start up. Sorry about that. There we go. So that's the way it was describing it. It kind of mentioned so a couple little additions. Let me see. Let me share. Let me back up for a second here. Let me back up. All right. So one of the things that it mentioned, it mentioned a little bit of. Hold on. Sorry. Oops. We don't want that. All right. Okay, hold on right here. All right. So the way it kind of described it, and everyone describes it a little bit differently as far as the details. The main thing to recognize is that you've got signals that are coming from the bladder. And here, actually, as you can see right here, from the bladder, it goes to the spinal cord. It didn't describe going up initially. It just described it happening here. But what's actually happening is it goes from the bladder up the spinal cord to the pons. That pathway controls all the involuntary stuff, right? The involuntary stuff meaning, for example, the detrusor muscle and the internal urethral sphincter. As far as the voluntary stuff, the need to go and the control of the external sphincter, that's where the cerebrum comes in. And that's where, of course, cerebrum is kind of conscious thought. That's where, for example, whereas this nerve was normally keeping this sphincter closed, conscious thoughts and need to go will actually inhibit that, which will allow it to relax and enable us to go. So that's kind of how that works. Hopefully it's not too confusing. So, all right. Well, thank you to the 6 of you for showing up early. Any questions at this point with anything we went over?
No.
Okay.
Is there
All
any?
right.
When is the extra? When will you grade the extra credit?
Oh, so the extra credit I think I put down is due like on May 27th. If those of you that have sent it into me, I probably will have it within the next week. At the latest.
Okay.
Yeah. All right. Well, no other questions. I'm going to go ahead and I've got this recorded, so I'll go ahead and send this to you. Just remember that both exams now are on June 1st. Thanks for letting me know about that. And I will see you on Wednesday for the reproductive system 1. Like I said, I need to update that lecture. So have a good day, and I'll see you on Wednesday.
Thank you.
You're welcome.